Hello students, this is Dr. Faizan Mirza. We are discussing control and coordination in plants. Just like mammals, plants also carry out control and coordination electrically as well as chemically. Here I am discussing with you the electrical coordination and communication within the plant. Let's take an example of Venus flytrap. Venus flytrap is this kind of plant. It is having two lobes. The two lobes are having stiff hair at the outer ends and there are sensory hairs inside the lobe. Now these stiff hairs, they can interlock very much like the catcher that you actually use for just assembling your long hair. So these are again of the same sort. These are stiff hair, and these two lobes are having three sensory hair in each lobe. So the first lobe is having three sensory hair, the second lobe is having three sensory hair. So first of all, you need to keep in mind there are two types of hair: the stiff hair and the sensory hair. The stiff hair is present on the outer edge, the sensory hair are present inside the lobe. Now, these lobes, they produce nectar here, and when nectar is being produced here, this Venus flytrap plant, this plant is of size of these lobes together, they are the size of a closed fist. So the insect will come and it will land on this region to feed on the nectar. When the insect has landed here and it is feeding on the nectar, by chance it will, as it is moving, it will brush against one of these three sensory hair or one of these three sensory hair. The moment it strikes one of these sensory hair, this allow the sensory hair to deflect. Now, if a second sensory hair is deflected within 20 to 35 seconds of the first sensory hair that got deflected, calcium ions will flow inwards into these lobes. Action potential will travel all across the trap and the trap will close within 0.3 seconds. As the trap gets closed, these stiff hairs, they interlock. When they interlock, the insect gets trapped inwards. The insect keeps fluttering. The trapped insect keeps fluttering, and the further fluttering of this insect stimulate these sensory hair even more, and the trap stays tightly shut. The glands here will then start to release certain enzymes onto the insect, digesting the insect altogether. And once the insect has been digested, so obviously the insect would have died by then, the fluttering would have stopped, and the lobes will open again to just wait for another insect. If the insect is very tiny, the insect would brush against this particular sensory hair and it would take a lot of time to reach to the next hair. The time of 20 to 35 seconds allow this trap to ensure that it doesn't eat or feed on very tiny insect, because that would result in a lot of wastage of energy, because it will be spending too much of energy and consuming too little of the nutrients it will be getting from the tiny insect. But yes, if the insect is large, then obviously there is no chance that the insect is going out, because the trap will close within 0.3 seconds, because at least two sensory hair would have deflected within 20 to 35 seconds because of the movement of the insect. How the action potential is generated in a plant cell, there's certain difference here. We know there was cell wall, the cell membrane. The cell membrane of a plant cell is having relatively negative potential. This is the resting membrane potential. The resting membrane potential is lost not by moving in of sodium ions, but by moving out of the chloride ions. Now this is the differentiating feature. In mammals, in animal cells, we have studied that the depolarization occurs because the sodium ion enters the cell. Here the sodium ion is not entering, rather the chloride ions are leaving the cell. We know that the membrane was negatively charged. If negative ion is going out, again this means that the membrane becomes depolarized. So the depolarization occurs, this triggers action potential. The action potential crosses this cell and reach the next cell through the plasmodesma, and it reaches the adjacent cell and then the next cell, and the action potential keeps on traveling way slower than the neurons, but it still travels. How the membrane will recover and regain the resting membrane potential? It recovers the potential by sending out potassium ions in the way in which the mammalian cells do. That's quite similar to what we do as well. That's it from my side. Thank you so much.